Professor Mohamed Banni. He's professor of molecular toxicology at the Irish Institute of Biotechnology, Monastir, Tunisia, and is responsible for a research group that work on a toxicological aspect in relation to the effect of contaminants, emerging contaminants on cellular function and molecular processes. He have a large scientific production, and he's visiting professor at the University of Medicine Vanvitelli, Napoli, and UPEC Paris. And he is moreover coordinator of 2 current European projects. And in addition, you are a big friend for me. Thank you very much, Mohamed, for being here with us, and the floor is yours.
Thank you very much, dear Margherita. Grazie mille per l'invito. Sono veramente molto contento di essere con voi. Ti ringrazio del cuore per questo invito, anche Gea per essere sempre carina. So probably now I switch in English, and my presentation will be about microplastic from the environment to human health. And as you will see in this presentation, the first part will be how far micro and nanoplastic could impact the environment. And then directly, I will try to make the link between the environment and the human health through 2 studies that we are performing on cell culture of human cell culture. So if we are here now just trying to speak about plastic, because plastic was reported to be present in all the components, first of all in the marine environment, but not only in the marine environment. As you can see here, you have plastic on salt that we use for our daily routine eating stuff. Plastic was reported to be in the water, in bottled waters, and recent publication of Margherita Group also confirmed it. Unfortunately, we are struggling against some lobbying, trying to demonstrate that plastic is really present, but not only visible plastic, because some organizations are taking care about the visible plastic, which I consider the minimum disastrous effect. But the micro and nanoplastic, the invisible fraction, would be the most harmful and should really keep our attention. So my group, during the last years, we published a set of papers on the presence and impact of microplastic on marine component, either fish or sea worms. And these reports indicated clearly that there is a real impact of this microplastic and nanoplastic on the biology of these key organisms in the environmental composition. I will jump because of time restriction, but we first demonstrated that plastic could be uptaken and then distributed and then, let's say, located in cells in fish. And the first study was on sea bream, branzino in Italiano, the sea bass that is a key species for the Mediterranean consumers. And unfortunately, we are facing this highly level of uptake of plastic in the environment that will lead finally in our grilled fish. The same thing was demonstrated for sea worms and their fundamental and pilot role in the degradation of organic matter in the sediment, and at the same time, exposure to plastic that will highly impact this microenvironment. So yes, plastic and agriculture, it sounds a little bit strange, but we were the first to publish, along with Margherita Group, the presence of nanoplastic in edible fruit and vegetable. We are eating carrots, we are eating potatoes that are sometimes highly impacted with microplastics. And my group recently published the effect, the combined effect, which is one very important aspect, which is the Trojan horse effect, or the plastic ability to veiculate toxic elements into living organisms. And this is the first one at the scientific level that published the the possible role of plastic in vehiculating herbicides. And this is the 2,4-D, the most important one, along with glyphosate, to be used in European and North Mediterranean countries. Yes, this is my best, topic, demonstrating that plastic could be a vehicle of toxic contaminant or not, the so-called Trojan horse effect. We did the same with nanoparticles, titanium nanoparticles or metal nanoparticles, and the result was not really definitely, concluding. But with plastic, we have a lot of evidence that the plastic could vehiculate this kind of contaminant, either, hydrocarbons like PAH or also heavy metals, but not only. My best, this is one illustration that could give an idea that what we are talking about in terms of plastic is a cocktail. A mixture of many pollutants that are all together in the same final matter. And this is one aspect that I have to defend, because many research groups are using polystyrene, for example, to expose their organisms, and then deliver some data on the effect of plastic, leading only on polystyrene alone, polyethylene. But it is not the real fact in the environment. In the environment, we have a composition of plastic of various size, different type, different forms, and also with inputs of other chemicals. So the toxic effect that we will observe in the environment will be completely different from the one we are challenging in the lab. So my way to do research now is to use only environmental microplastic that I can collect along beaches, along agricultural coasts, agricultural soils, and then prepare these micro nanoplastics for exposure in laboratory condition. And this could be in somehow near to the realistic events. It is my approach, and I think that it could be also something that we can rely on. And this is one very nice publication published this year, 2 months ago, on Journal of Hazard Material, with the impact of environmental microplastic alone or combined with benzoypyrin on the muscle mitolus galloprovincialis, hemocytes. And the same was published also on environmental pollution, along with the colleague from Messina, on the sea worm, and where we demonstrated clearly that plastic can vehiculate the pollutants from the water. One of my best slides that I did along with my dear friend Eduardo Pugliese here present is this one. And this paper published 2 years ago was the first to determine and to give evidence of the presence of biofilms on floating microplastic. The plastic was collected from the open sea, and then with SAM technique, we demonstrated that on that plastic, we have a variety of bacteria that may be vehicle by mussels when filtering water for feeding. And this data was also correlated with metagenomic on plastic, demonstrating that in some case, we can have, and it was the case in this study, some pathogen strains of bacteria like Vibrio fischeri that should not be present really in the seawater. So this is a real proof that plastic may be transferred from the environment, vehiculating other component to human. And now switching to human, are two very nice publications that I did along my group with Margherita group. The first was micro nanoplastic in edible fruits, performed in totally here in Catania with using material from Catania. And the second is the presence of microplastic in edible fish. And it was, if I remember well, the sardina, the sea bass, the mussels, and so on. Just for the history, these two papers, we struggled enough to make them published, because high-ranked journal, I will not say refuse it, but it was hard to convince them to publish this data, even they are really very, very, very interesting, but probably they are not well accepted by some lobbies. I don't know. And this is to stress this fact. This is a paper published on Nature, the most important, let's say, journal in our field. And the question, and this is 1 year ago, the question was, microplastic are everywhere, but are they really harmful? You know, the way you ask the question can give you also the answer. I mean, when you ask, but are they really harmful? It is a way to say that probably not. And I was really very shocked when I read this paper, because it is a certain way to say plastic, probably they are in the environment, but we are probably exaggerating, which is not the case. And I will show you now, right now, proofs of that. So our idea was to take bottle of waters, to ground them, and to produce a particle smaller than 2 micrometers and smaller than 1 micrometers. And this plastic particle of polyethylene terephthalate that are really present in the water that we are drinking every day. If you are drinking water directly from the bottle, you are drinking around something like 5,000 microplastic with size less than 3 micrometer. These are real data. So we produced either polyethylene or the polyethylene terephthalate, and it was in collaboration with Margherita and Gea. And these plastic particles were there used to expose either OH22 cells from liver cells, but also stem cells. And the data that I will present here are really, so the first are the human hepatocellular carcinoma cell lines, cancer-derived cells, but you will see the data that I'm presenting now. And the second are the mesenchymal stem cells, either from bone marrow cells or adipocyte cells, 2 components of our human stem cells. And we made just the exposure of the cells to polyethylene terephthalate and to polyethylene. Well, I will switch. Okay, this is very interesting data, because if you can see the first graph for OH22, the size is determining the toxicity of plastic. I mean, the more the plastic are small, the more the toxicity is, and this is determined by the calculation of the EC50. And this should give the answer that what we are, and I agree with Gea when we discussed that what we are observing under the microscope when we are monitoring plastic are those that are retained on the filter. But the most toxic are those who are passing the filter that we are unable to evaluate till now. So the toxicity derived from the smaller particles, because probably they are more easier to go inside the cells. Yes, we are talking about OH22, cancer cells that here should not die very easily. And our results, first result, demonstrated that exposure to environmental microplastic are decreasing the ability of the plastic to the cell to die. So the final data that I will show you is as if the exposure to plastic are giving more carcinogenicity to these cancer cells, making them more resistant to death and probably providing something that will alter their capacity to undergo either apoptosis or autophagy. So yes, a lot of oxyradicals that are produced inside the cells. But when we decided to study autophagy and apoptosis through 2 markers of cell death, and with targeting very important protein involved in the both processes, we showed that, for example, BAX, BCL2, which are the 2 proteins that could initiate the apoptosis, and mainly BAX, who should increase in the cell, we have the opposite result. I mean that the cell are not undergoing death. And this is a very harmful and wandering result. So I will just, for time restriction, go to these nice pictures, which are immunofluorescence, and we are trying to localize the presence of protein involved in such process, like here P62, which is very involved in autophagy. And as you can see here, P62 was lowered in presence of plastic. That means that no autophagy, no death. LC3 also very involved in autophagy, and the same was observed because LC3 and P62 are the opposite trend. So here we are in an increasing trend, meaning that these cells are refusing to die. The same with other markers like BCL2 cell lymphoma 2, etc., etc. And I will just scroll on because they are very nice to show this data. And I will arrive to the conclusion after all this experiment with caspase, for example, here, caspase-3, which is the protein who will execute the apoptosis. Here we have something that really it increased, but without really a significant effect. Okay. Cytochrome C also, MCL1, 2, but my conclusion will be that microplastic increase cell proliferation. It is the opposite of what we are supposed, a toxic component is supposed to do. Microplastic inhibited either apoptosis or autophagy. It increases the level of ROS. So our final conclusion is that microplastic is probably rendering and transforming these cancer cells to more cancer's capacity. And this is very warning. I mean, for normal cells, it is really something that should be taken into consideration. So this is the first data. The second data is this one, and these are stem cells, cellule staminali. And these stem cells are exposed to a microplastic, either from the bone marrow or from the adipocyte cells. Just for the information from the bone marrow, these cells should undergo differentiation to provide osteoblasts through chondrocytes and then our bones, or adipocytes. And there is a certain kind of balance to in harmonized subject. So here we just observed that the number of cycling cells undergoing divisions are reducing drastically. That means that plastic with the size less than 1 micrometer are blocking the division of these cells in such a way. The second one, using flux cytometry, we observed that the percentage of cells in S phase, the phase where the DNA is synthesized, is really decreasing. That means that these cells neither produce new DNA for division, neither undergo divisions. And this is the direct effect of polyethylene terephthalate. And the level of senescence of these cells are increasing drastically. And this is really very warning. I mean, for this kind of cells that are there to support the renewal of the body cells. Apoptosis 2, if you see the second graph, you will have the evidence that with polyethylene terephthalate with size 2.6, we have an increase, a drastic increase in apoptosis event. And you can see the difference between carcinoma cells and normal cells. Yes, these are key proteins that are involved in apoptosis and autophagy, and mainly the P51, P57, and this is an upregulation of the principal cytokines involved in cell cycle arrest, as by P27. That means that we are really undergoing apoptosis through the arrest of the cell cycle and then the apoptosis event. Okay, and then this, one of the best data of this research, that the effect of polyethylene terephthalate on the stem cells stemness. I mean, which kind of cell will be produced if the initial cells are exposed to microplastic? Our data showed a shift toward adipocytes. I mean that exposure to environmental microplastic are shifting the way of differentiation to adipocytes respect to the blastocytes, to the osteoblasts, sorry. And these are some specific stains showing that adipocytes are more present than osteoblasts when original cells are exposed to environmental microplastic. Okay, the same evidence, but here with the capacity of the cells to differentiate to adipose, osteo, and chondrocytes. I mean, the same data, but with the other markers related to the gene expression of key proteins, of key genes involved in this process of cell differentiation. I will go very fast to avoid you the loss of time. And here, for example, the beta-gal test also is in favor of a harmful effect on this kind of cells. Yes, staining with X2AX and ATM specific stain showed that because the first one is a key regulator of cellular response to DNA damage. The second is a kinase that regulates DNA repair. So if the ability of the cell to make reparation or not, or undergo apoptosis, and our data are clearly in favor of apoptosis more than reparation of DNA or DNA repair. Okay. So to conclude with this second part, the translation from the environment to the human health provided us clear evidence that microplastic modified the fate of these stem cells in vitro, including senescence in these cells with loss of several properties of the stem cells. And this paper we were also really struggling to publish this paper despite the very high level quality of the research, because I think it is a problem for editors to publish such data. But finally, we published this very nice paper, and it was 2 weeks ago accepted on environmental research. And I really should thank Jose Domingo, the editor of the journal, because he took his responsibility as editor of a higher ranked journal to publish such data. It is not the case for many others. So, and this paper is a very nice collaboration between my group, a group in Sergio Minucci in Napoli, Università Luigi Vanvitelli, and also Margherita Ferranti group with Gea. And finally, I have to thank my group in Tunisia, who are really doing a very nice job with the high level production. And thank you for listening and for sharing this moment with this research topics. Thank you very much, Margherita.
Thank you very much, Mohamed. Our collaboration is very important for me, and thank you for your time for us. And I hope in the next future many other work together, Eduardo, together Moreida, and the Messina group that are my important friend. And I hope that this work is useful for the community to change this important warning topic.
Yes, thank you very much.